Hey guys, this is Mr. O'Brien again, and this is the second part of your Themes of Life PowerPoint. Now, in this portion of the PowerPoint, we're going to be comparing cellular structures and their functions in prokaryotic and eukaryotic cells. So, let's first kind of go over what the difference is between prokaryotic and eukaryotic. A prokaryotic cell would be one that is located down here. Notice they're smaller, they're less complex. Prokaryotic organisms are ones that do not have a nucleus or membrane-bound organelle, so they don't have any mitochondria, no Golgi bodies, no endoplasmic reticulum. They're much, much simpler. Now, prokaryotic cells, they actually reproduce using asexual reproduction. And what this means is they make clones, or they make an exact copy of their DNA. So these clones, they are extremely similar, and the point of this is to make a large number of individuals over an extended period of time. Think about when you're sick. Bacteria want to keep replicating and replicating and replicating until they kind of begin to start making you sick along those lines. Second type of cell that we're going to talk about today are eukaryotic cells. And eukaryotic cells are ones that contain a nucleus as well as membrane-bound organelles. So when we talk about a nucleus, we're talking about this structure right here. And let's do a different color. This structure right here. So this structure right there, that nucleus, this is where our DNA is stored. So DNA in prokaryotic cells are just kind of floating freely in this nucleoid region. There's no membrane surrounding it. So that's the big difference right here. A eukaryotic cell contains a nucleus. Prokaryotic cell do not. Now, prokaryotic cells, let's talk about what they do contain. So a prokaryotic cell contains DNA. Now again, all organisms, remember, contain genetic information in the cell. But if you remember, this DNA is just kind of floating freely on the inside of the prokaryotic cell. There's no membrane enclosing it. They also contain a plasma membrane or a protective barrier on the outside. That would be this area right here. Again, this is made of phospholipids, and this protective layer kind of just keeps everything in place. Ribosomes are also present inside of here, and ribosomes are, remember, where our proteins are made. And remember, all organisms need proteins, and these instructions for the proteins comes from our DNA. And the final thing that's found in prokaryotic cell is this flagella right here. And again, the flagella is used for movement. It's almost like a whip-like structure that's kind of used to move it through a liquidous environment. Now, eukaryotic cells are much more complex, and that's kind of the big thing. Eukaryotic cells are complex, and if you look over here, look at all the different structures that are found inside that cell. The big thing that we have is the nucleus. Remember, the nucleus is where our DNA is stored right there. Now, we're not going to talk about all the organelles, but they have membrane-bound organelles inside of them. So that would be things like this right here, the endoplasmic reticulum, which is actually used to ship things to the Golgi apparatus. It modifies proteins and ships them to the Golgi apparatus, where another modification occurs and it's shipped out of the cell. We also have mitochondria, which is where our ATP is produced, lysosomes, which break things down, and there also are some ribosomes as well. So again, eukaryotic cell contain these membrane-bound organelles and membrane-bound nucleus. All eukaryotic cells also have a plasma membrane, which is the outside layer made of phospholipids of the cell. Again, this is found in both eukaryotic and prokaryotic cells. And finally, ribosomes. Again, ribosomes make our proteins in the cell. So again, kind of to throw this into perspective, the nucleus contains the instructions for proteins. The proteins are made on the ribosomes. The ribosomes are modified in the endoplasmic reticulum and then modified again and shipped out of the cell in the Golgi apparatus. So let's talk about some differences between prokaryotic and eukaryotic cells. So one of the prokaryotic cells you're probably most familiar with is bacteria. Now, this is a blown up picture, of course, but bacteria are microscopic. They're so tiny that we need a very, very powerful microscope in order to see it. Now, prokaryotic cells are smaller. I'm going to highlight these. They're smaller as well as less complex. Again, remember from the last picture, they're much more simple. Eukaryotic cells are cells that are found in plants of animals, like our toucan over here. Again, they create a more complex organism. So, eukaryotic cells contain a nucleus as well as membrane-bound organelles, and they're much more complex, and they form more complex organisms. So again, if you see a cell and you see a nucleus on the inside, you automatically must think that is a eukaryotic cell. So there still are some similarities between prokaryotic and eukaryotic cells. And the first one is all cells use ATP. Now remember, ATP is adenosine triphosphate. And this is the energy currency or the energy source in every single one of the cells on Earth. So every cell needs ATP to survive. It needs to produce this ATP to do all the cellular functions it has. All cells also contain ribosomes because all cells need proteins. All cells have a plasma membrane. And remember, plasma membrane are made of phospholipids, which contain a head and two tails. And usually this plasma membrane makes what's known as a phospholipid bilayer. So these two layers of phospholipids, they are arranged like this because the heads want to be associated with water and the tails are hydrophobic. Also on the inside of the plasma membrane is this liquid that kind of holds everything in place. I like to think of this as almost like a Jello mold, right? Now inside the Jello mold, sometimes we put pieces of broccoli, pieces of fruit. I'm going to draw it as a green-orange, but you get the idea. And all of these things are held in place by the Jello. That's kind of what the cytoplasm does. It holds everything in place, allows for cellular processes to occur. Now, let's draw some of these cells. And the first cell we're going to draw is a prokaryotic cell. So a prokaryotic cell, again, is very simple. We're going to draw a simple version of it right here. Now, that black layer that I just drew, again, was my plasma membrane, the outside covering of the cell. We also have on the inside a circular piece of DNA. And again, remember, there's no nucleus, so it's just kind of floating freely on the inside. All right, so there's our DNA. We also have ribosomes. And ribosomes, again, is the site of protein synthesis. And these ribosomes are kind of floating freely in this liquid called the cytoplasm. So all of this space here, all of that is liquid. And we call that the cytoplasm of the cell. And finally, we have a structure, a whip-like structure that is used for movement. We call that the flagella. So again, all of these things found inside of a prokaryotic cell. And you'll notice this prokaryotic cell is simple. It's not very complex. It's simple and it's small. Now, plant cells are a little bit different. So plant cells are a little bit more difficult to draw because they actually have two layers. They have a thick outside layer that is used for structural support, so trees can grow big and tall. And that outside layer is called the cell wall. Now, this is different than animal cells because animal cells don't have that cell wall. Remember, they have what's known as a plasma membrane. And that plasma membrane is actually on the inside of the cell wall. And this cell wall actually has holes in it so things can go in and out. So again, plasma membrane. So inside of plant cells, we have a couple different structures. And the first one I want to talk about is obviously the nucleus. So the nucleus is where our DNA is stored inside of eukaryotic cells. Notice it has a membrane, and there's also this little tiny darkly stained area called the nucleolus. But this is where the DNA is stored, called the nucleus. Now, usually associated with the nucleus is another membranous structure. And this membranous structure is called the endoplasmic reticulum, or ER for short. Now, we have two different types of ERs. One of them has these little tiny bumps on it, and those bumps are actually ribosomes. So we call that one with the bumps the rough ER. The ones that don't have bumps are called the smooth ER. Now, both of these structures are used to transport and modify things and move them within the cell. The rough ER usually transports and modifies proteins, and those proteins are usually sent to this flattened disc-like structures. Kind of looks like a stack of punched pancakes. There's usually some vesicles budding off of it. And what happens is the proteins get shipped to here, and the final modification occurs, and they are shipped out of the cell. These are called the Golgi bodies. Again, final modification before they're shipped out of the cell. Now, there are also some ribosomes floating around freely in the cytoplasm, or the liquid. Again, this is where our proteins are made. They are also located on the rough ER as well. A specific structure to plants are this very large storage mechanism called the central vacuole. So this stores things like food, water, and the reason that plants have this is because they can't move, they can't go and eat something, so they have vacuoles to store all of that stuff in there. Another thing they have, a very important one, is where photosynthesis occurs. And you'll notice that inside of these, there's usually these flattened discs. And this is actually what absorbs the sunlight for photosynthesis to occur. And again, this is called our chloroplast. Again, chloroplast is the site of photosynthesis. Now, a lot of people get confused and think that plants don't perform cellular respiration, but actually they do. They still have mitochondria because they need they need ATP to be produced to perform cellular structures. So the mitochondria usually has these folds on the inside, and that's kind of how you can identify them from the chloroplast. So again, the mitochondria, this is where ATP is produced for cellular activity. So that gives you a pretty good representation of a plant cell. And this is a little bit different than an animal cell. An animal cell doesn't have that rigid wall on the outside. It only has a plasma membrane. So the plasma membrane is still the outside protective layer, but animal cell, no cell wall. So the animal cell obviously has a nucleus because it's a eukaryotic cell. That's where our DNA is stored. So again, our nucleus identifies it as a eukaryotic cell. So also on the inside here, or just outside the nucleus, I should say, we also have this membrane area right here. And again, remember there's two of them, a smooth and a rough ER. So this again is our endoplasmic reticulum, which modifies and ships things within the cell. So this is our rough ER because ribosomes are attached, and this would be our smooth ER because there's no ribosomes attached. Now remember, once the proteins are made on the rough ER and modified in the rough ER, they're sent to this flattened pancake-looking membranous sac where the final modification occurs and shipped out of the cell. This, again, is our Golgi bodies. Now, again, if you were paying attention in the last one, there's also these structures floating in the cytoplasm or the liquid of the cell where proteins are produced. They are also located in the rough ER called the ribosomes. And we also have one of the most important structures in animal cells, and that is the mitochondria. Now remember, the mitochondria has these folds to increase surface area to increase ATP production inside of our cell. So that should give you a pretty good understanding of the difference between plant, animal, and prokaryotic cells. Remember, prokaryotic cells do not contain a nucleus and do not have membrane-bound organelles, whereas eukaryotic cells do. Again, a little bit different between plant and animals, but that should give you a pretty good idea. Hopefully this helps, and we'll continue with topic 1 a little bit later in the class. Hope this helps. See you later, guys.